Next speaker, the last speaker from this section, is Dr. Maurice Whelan from a European Commission Joint Research Center in Italy. He's going to talk about facilitate the uptake of the in vitro methods in research and testing.
Thank you, Chair. Good afternoon, everybody. We're still in the. It's lovely to be here. Thanks so much for having me. Yes, I work for the European Commission Joint Research Center. And you're probably wondering what I'm doing showing this slide. Well, actually, this was my first encounter with phenotype investigation. And it was actually an Irish artist who visited me in the Joint Research Center some years ago. And this is her artwork. She was fascinated with exploring through artistic medium phenotype in people. And at the time, we were looking for a reason to collaborate. We couldn't link the in vitro work that I was doing with interests that she had, so it kind of fizzled out. And I brought her book. She left it as a present. And we probably could construct a quiz later. Can you, all experts, identify her molecular sculptures? She's up to the back of the... Yeah? Here it is. I don't know how to get it even closer. It's practically in my mouth at this point. Do you want to clip it to my shirt, maybe? I think it might even get too close. Is that how does that go? Is that better? Okay. So where was I? Yes. Challenge for you all to see later. Can you go through the pictures and see, can you identify? Because her sculptors are made actually out of the straws they use for artificial insemination. So constructed those. So see, can you match the traits of the cattle with the molecular structures or sculptures that she... So years went by until last July, I think it was. I had the pleasure to meet Elisabetta in Milano. And she started talking about this genotype to phenotype. And I went, oh God, is this coincidence or is it destiny? So I had to, of course, accept this kind of invitation to come because but look, I'm a bit the outlier talk today because I'm just going to be giving you a general, information about who we are and what we do. And hopefully that will trigger some ideas for potential collaboration and so on going forward. Those who aren't familiar, you know about the European Commission, I guess, but within the Commission we have the Joint Research Centre. We're the in-house knowledge and science service of the Commission. So our job is to provide scientific evidence and advice, impartial, to policy making in the EU. So we work closely with our policy partners in Brussels. I'm actually based in Ispra in northern Italy, but the JRC is a big organization with about 2,000 people on our site in Ispra. You can watch a little movie. So we support all different policy areas. One particular is the policy in the EU that we have for protecting the welfare of animals used for scientific purposes. And that's, as you're probably aware, within that directive, that sets out all of the structures for getting project evaluations and ethical approval and so on. And in that, the directive sets out the duties for the EU Reference Laboratory for Alternatives to Animal Testing, which is the Joint Research Centre, and I'm responsible for that too in Ispra. As you can see, we have 4 primary duties: to participate in research and development. We do a lot of work in validation of methods, particularly for use in regulatory toxicology and safety assessment of chemicals in different sectors. We work on dissemination and promotion of alternatives, and that's one of the reasons why I'm here. We have other important policy areas coming as well. I mean, it's clearly looking at the work that you do. That's critical for all kinds of policies: our agricultural policies, our competitiveness policies, our food security policies. So you know, this aspect is just one policy of in terms of protecting the use of animals in science. And the Commission also, we're working on defining a life science strategy for Europe, which will be out in the summer. A lot of focus on biotech and aspects, both in health and agriculture and so on. So that would be an important framework. I'm personally very involved in defining that. We have a Biotech Act coming out next year from the Commission again, would have a lot of relevance to what you're doing. And also we have a roadmap to phase out animal testing in regulatory testing in chemicals, pharmaceuticals. So these are our other policy documents coming out. We operate a network of validation laboratories, 33 laboratories around Europe, mainly CROs, expert in vitro. Again, tailored towards the validation of methods towards toxicological testing. But I know, you know, you're interested in infrastructures, and I'm really interested in infrastructures, and that's something we maybe learn from each other. We publish an annual status report, we call it. Basically describes a lot of things that we're doing ourselves, but also in collaboration with partners. Next version is coming soon, but you can download it. It's easy to, you know, glance and read, and you might catch your interest. We also run a summer school for primarily training early career scientists. Our next edition is in May. We run it every 2 years. Unfortunately, the applications are closed, so but maybe in a couple of years' time, it would be very interesting to incorporate this topic potentially into the summer school, and that would be really exciting for us because we're always looking to branch out. And it's great. We have 120 enthusiastic early career scientists on site for a whole week, expert speakers who stay the whole week. We like this kind of setup, which is really, really wonderful. And of course, it's very informative and stimulating for us to learn a lot. We compile statistics on the use of animals used in science in the EU. That's part of the directive as well. Each member country has to report uses, and we actually, my group compiles these statistics and produces these kind of graphs. We don't need to look into everything. But basically, we're using about 8.5 million animals within EU27 in Norway. You know, nearly 3/4 are used in research. Actually, not so many percentage-wise are used in regulatory testing, and a big focus on human-centered research. Curiously, before I came here, I was thinking, okay, you know, Eric's talk this morning, that kind of in vivo phenotype to phenotype research, where does that fall into this kind of use of animals? And there isn't a specific category. Probably falls into the genetic use of genetic engineered animals, and something maybe I'll be curious later to see when you're questing, permission to carry out these studies. How would France, for example, categorize? How would it report use of animals? So, we've been very good in Europe at measuring and recording the use of animals in research. But 7 years ago, you know, particularly talking to people in the parliament, and there was no real sense about, well, how much are we using non-animal approaches in research? And it was all kind of anecdotal. We did an analysis of the literature for the last 10, 20 years. It showed that around 2016, the number of, you know, biomedical science-related publications using non-animal approaches exceeded the number of publications using animals. So that was the 2016 as a kind of a turning point. So we said, you know what, let's do more systematic analysis of the use of non-animal models in research and get a sense of, you know, the scale of what's going on. Of course, that also allows us to see what kind of models are used, what trends are there, how, etc. So we did it. It was human focused, focused on 7 disease areas. And you can see, you know, it was over 300,000 abstracts, over nearly 100,000 texts. But in the end, actually, what we found is there's a great deal of non-animal models being used in biomedical research. We identified over 3,000 models in these areas. These are you know, stand-up models. They just weren't anything. They had to have some pedigree, let's say. By the way, we published what we call executive summaries. These are just 3-4 pagers describing generally the findings, targeted more at non-scientific experts, get a general sense of what's out there. We then have a technical report that describes how we did all of this, much more detail. And then, of course, we have a catalog of all the methods that we found, described in a very systematic way, and that's available in the catalog. And of course, we hope that researchers like yourselves who might be interested in a certain type of model or something will be able to access that kind of scientific metadata that you need to then go and study the publication. But also, more generally speaking, the higher level documents are quite informative. But what it allowed us to say is there's a lot going on out there, and it's not just an emerging field. It's a very, very established field, and there's a lot of serious research being done with non-animal approaches, as you well know. Look at your own research. Some general findings, not surprisingly, different research areas prefer different types of models depending on the type of research question you're looking at. For example, in cardiovascular area, in silico computational models are used quite a lot, which you can understand as modeling the whole physiology and biomechanics. Big dependency on using, cell-based systems. A lot of, using immortalized cell lines, cancer cell lines traditionally, but going much more towards now iPSC and so on. And we're seeing a steady increase in organ and chip. In fact, that's where I met Elisabetta, was at the European Organ and Chip Society meeting in Milano. So, again, okay, it's human health, it's human diseases, but when you look at how these models, are built and how they function, there's a great opportunity for translatability, working these models into your own research. This is what we're working at the moment, because that was a very laborious effort to do that. You can imagine, hundreds of thousands of things and loads of contractors working. But with the emergence of AI and, large language models and text-based tools, we've now started this project which the Parliament actually is funding to make this much more a dynamic process. So not only are we applying this now to update the disease areas that we were looking at, but also add in new disease areas. Really importantly, we're going to make the tool available so that people now can, because the future isn't static databases and big catalogs that everybody updates, once every few years. The future is, creating catalogs and databases on the fly using these tools. So with this tool, we hope that communities like yours will be able to customize it and build your own databases and catalogs and so on. That really is tailored for your needs. And we'd like to interact with the community that we haven't been directly serving, see how translatable these tools are. I mentioned stem cells and iPSC, and we all see the importance of that as a research tool. From our side, being the commission, we're not developing new technologies or searching for new knowledge per se, like you are. We're looking to see how we can support the emergence of new tools and the impact of new tools in research communities. This was a conference back in the summer, session that we organized with the International Society for Stem Cell Research. The focus there was acknowledging that there are actually a lot of promise, huge excitement over these tools in research, but can we stand over the quality, the reliability of the results that we're producing? We're producing a lot of results very quickly and publishing a lot of papers, but is it trustworthy? And we really, as scientists, use other people's results and work. So this discussion was about really acknowledging that there are a lot of good practice and standards out there, a lot of awareness of what constitutes good practice and standards. But are we really implementing it? And how important that might be. There's a manuscript that actually has just been accepted. It's just under a final review. It's going to come out in Stem Cell Reports. And Lucia and Milena are in my team. They're co-authors or lead authors on this. And these are the kind of recommendations that are coming out. But look at the main finding. maybe it's not a big surprise to you, but you know, we need to do better. We need really a cultural shift towards prioritizing quality and reproducibility. Now, I know as a young researcher, I remember these are like your parents telling you to stop showing off or something. You know what I mean? It was like, uh, but seriously, you know, we need to think seriously about it, and it's a really a collective responsibility. You know, we've involved journal editors and so on looking at policy, and that's a challenge for journal editors because, you know, in one hand everybody agrees, oh yes, we'll ask the author guidelines, we'll ask for this and we'll ask for that, we'll have to, but then people say, I'm not going to publish in that journal. It'll take me, you know, twice as long to write the manuscript than if I go to another journal. So there's a trade-off there. Or facilities, and again, I look to the infrastructure that you're building here and plan to build in the future. Massive opportunity to implement standards, raise standards, you know, ensure quality and reproducibility and reliability through workshops like this as well. Training, very important, you know, the access to reputable suppliers of materials, you know, sustainable supply of materials. You know, many of the research that is published, you know, you could never be reproducible because you can't actually get reagents or the cell models or whatever they were using, etc. So again, maybe not all that sort of new, but really a call to action to the community to. It's not so much about we don't know what good practice is, but we should start practicing what we preach. Another area that we've been working on a lot to improve, again, translatability, knowledge sharing is good reporting. Nothing new to you, I'm sure. Many times, I'm sure, when you try to reproduce or somebody else's work, it gets super frustrating when you start getting down to the level of protocol and what did they do here and where's the secret ingredient and why did. And then it usually involves you having to contact the researcher and hope that they're available to you to, you know, even come and et cetera, et cetera. So, you know, we need to do better. So this is something that we, again, did with policymakers, funding agencies, journal editors, research associations, to really look and see how we can improve reproducibility, trust, transparency, et cetera. You know, again, recommendations for different parties. On the Commission side, we're looking and seeing how can we bring this into our framework funding programs. We don't want to overly constrain researchers and ask, you know, for too much, but how do we, you know, we're spending billions of euro on research in Europe, and it's not translating into innovation and competitiveness from an industrial perspective. And a lot of that is because the results aren't translatable, they're not trustworthy. So how can we really improve the quality of our outcomes? But again, you see recommendations for researchers. Simple things like the responsible shortcut citations. And many times you go out in the field and say, oh yes, I'd love to, I'm going to do this. And you go, okay, well, the material method section is really short, so it references something else. So you, okay, great. So it gives you an impression that all of the information you need to recreate this experiment is somewhere else. And then you go and you follow that reference, and it's another little material methods section that references some other thing. And then when you try to fact that there is no protocol and whatever. So, perfection is the enemy to good, but we can definitely do a lot better. We led this project at the OECD. Actually, I'm frequently in Paris, and the OECD headquarters just up the road. And this was to develop good in vitro method practice. It was again tailored towards more in vitro toxicological testing, but many aspects in this here would be very applicable. So I encourage you to have a look at that. You know, it's difficult to do this as an individual researcher. It's still important to be mindful, but definitely in terms of a laboratory and a laboratory team, you know, want to improve the quality of their output, but importantly too, pass on, you know, the next generation of PhDs, postdocs come through. You know, you're losing a lot of time and a lot of resources by making the same mistakes again, you know, and by not documenting things and so on. So these could give you a good framework on how to put those. Mentions the importance of standards of four. This was introduced by the Commission a couple of years ago, a code of practice on standardization. So, you know, research communities, knowledge communities like this, you know, we can do two things. One, we can put what we call science into standards. You know the technologies, you know the methodologies. What should be the standards? You know, that develop through process of consensus. So defining the standards should come from the communities that understand the methodology, technologies, and applications the most. Equally, putting standards into science, how much money is wasted through our research funding, where everybody keeps building their database and not complying to data sharing standards, are developing, you know, et cetera, et cetera. So we could make much better use of standards in science. I mean, look at all the talk today about all the genomics, genetic type, you know, again, are we using the right reporting standards, et cetera? So this is one particular, let's say, success story, something we started some years ago was organ on chip, and there's such a proliferation, and it's wonderful, of technologies and different models and applications. And, you know, we set out working with the European Standards Authority, then CENELEC, Standards Organization, to set out a roadmap for what would be standardization needs in this domain to support research and application. This was done very much in the context of the European Organ on Chip Society, EuroFAANG. And that roadmap has just been published and sets out priorities and how to go about it. Very much an inclusive process. And now that's triggered the creation of this ISO Technical Committee subcommittee, microphysiological systems and organ on chip. Now, this might sound all very administratively stuff and, you know, not something of interest to researchers, but this is really important. It's really important because this will set the standards, but it's the community that sets the standards, not invisible people. If you're interested in this, and we need people who know about the technology, the methodology, etc., you know, contact the French Standards Authority and ask them about this committee. And say, listen, I want to be informed. We, as a community, we want to make sure that standards that emerge out of this work fit our needs, you know, that they've taken into account our requirements and what we know about it. So, yes, I hope that will help ultimately get the... A lot of us that the technology, just wanted to finish on this. I read this back in 2006 or 7. 20 years ago when Sidney Brenner, he said, look, the real problem we have is we're getting to the point we can't speak to each other. We're all so super specialized in generating mountains of data and can't make sense of it. And that's the real, now he said this 20 years ago and it stuck with me. You know, it's a challenge in itself. As scientists, how do we manage our knowledge? We talk often about data and databases and data exchange and sharing, but that data just underpins information and knowledge, doesn't it? So as scientists, traditionally, how have we done it? And without being too philosophical about it, I mean, we often look to ontologies to be able to reduce the complexity of something into, you know, an organizational model. And we see ontologies everywhere in our daily lives and in our scientific lives. Go ontologies you're very familiar with, I'm sure. And even, you know, things like Google and all the rest of it are based on knowledge graphs, which are essentially ontologies. The knowledge community builds an ontology that reflects our understanding of the world. I started getting interested in about 2010, 12 in another new emerging ontology, what we call our adverse outcome pathways. And it was filling a gap, particularly in toxicology, because we were shifting from using in vivo, traditional endpoints, observational endpoints, apical endpoints, observing tumors in rats, for example, into in vitro mechanistic biomarkers. And bring it into regulatory use, the problem is we were observing and measuring on the left, but we're regulating based on what we see on the right. So we needed something to build this bridge between, you know, different levels of biological organization. That's what emerged as a new kind of conceptual topology, a multi-scale modeling. And it's very simple in one way. All we do is we talk about key events, molecular initiating event, again, from toxicological perspective, we're thinking about a molecule, an exogenous molecule interfering with a system. Response at cellular level, organ level, and then very importantly, a key event relationship, describing the factors that cause this transition, you know? So it's telling a good toxicological story. That's really where it emerged from. But then, because it was developed at the OECD, I just wanted to say, that's like a linear story. This happens, this happens, this happens. Of course, we know that many of these pathological processes share key events, inflammation or, you know, proliferation or something. And so, of course, you can build a network of those. You can say, well, a certain molecular initiating event could lead to multiple different types of adverse outcomes depending on context, on conditions, and equally vice versa. But this is the process now how we built it. So it's a nice concept, but actually this is a program, an international program of building this knowledge base. It's not trivial to build an AOP. There's guidance and methodology and invention to follow. Because building actually across different levels of bioorganization, it's very rare that somebody working in the molecular level knows a lot about the pathology, you know. So it's implicit, it's a requirement, work in this highly interdisciplinary, collaborative type of way. And of course, nothing's for certain. We have to weigh the uncertainties and see where there are gaps. And very important, it's endorsed. For me, this is, I think, the only regulatory endorsed knowledge base that exists. What that means is these AOPs are endorsed. Somebody then uses them in a regulatory testing context to inform some type of testing strategy, and that's considered, you know, good thing. And more recently now with this peer review is actually being done by journals. So this helps academics in particular, you know, contribute to developing AOP and the AOP knowledge base, but still getting the recognition of publishing in high impact journals. Go and have a look at the AOP wiki. It's all public, all free. It's a big community, and you can go and have a look and see. What we're really seeing is that community building happening. You know, people are beginning to come together and try to figure out inflammation or ROS, these, you know, things that have been around all the time, but really trying to codify it. And it's fascinating because you're getting people coming in or looking at inflammation in the brain or inflammation, and these communities start coming together and realize it's the same mechanistic process, but because they're calling the process or elements in the process by different names, they typically don't talk to each other. But then when you use this type of framework, you can really have that mechanistic. This was very successful. There's a whole series of papers. It's what we ran where people started seeing this mechanistic framework as something that can go well beyond toxicology. And actually, this was looking at there, of course, the molecular initiating event is, you know, viral infection, isn't it? Important here too was another aspect of the AOP, which was, you know, how come the pathogenesis was common? It was fairly obvious what was happening. Why was it affecting people in a very, very different way? So it was actually modulating factors around the AOP that really became the interesting aspect, and that's what's captured here. So I'm wondering, what about phenotypic outcome pathways? What about exploring if this kind of knowledge model, you know, would be a way of describing, you know, genetic traits, variations, whatever, with a phenotypic outcome? I encourage us to have a look and see. Just want to finish to say we do have laboratory facilities as well. You can do a 360 tour of our lab here. You see two centered on 2 high throughput testing platforms, and then in the back room there's imaging, and then we have organ and chip and other few bits and pieces. Actually, we've even created a virtual reality. Looks a bit more high tech than the actual lab, but we've been using that actually for training school kids in, you know, around with virtual pipettes and understanding cell culturing in a more rudimentary way. So again, if any of you are involved in school outreach, education programs, will be actually come online next week as a shared public resource, and you can run it on computers as well. You don't just need the. And yesterday, we've just opened a call for open access to our laboratories. This is to JRC because we're a public institution. We want to share our facilities. And there's a program for people to apply and come and spend time with us and use all our machinery and work with our staff and in a collaborative way. It's the first time we've done it with our lab. There are other JRC labs that have done this. We're the commission, of course, there has to be a bit of bureaucracy. So there's an application process, etc. But we want to make sure everybody gets a chance. So if you're curious, have a look and see if there's something that we're doing that might interest you. We can talk. Well, that's it. Thank you very much for your attention.